Hello, everyone. We are excited to share our research project developing an assay for the discovery of transplastic inhibitors. And for the last few weeks, our team has been working under the mentorship of Diego Logan to explore the unique transplasting mechanism in Ascaris.
This has been our project team for the last couple of weeks. My name is Tejaswini Anmareddy, and I'm from Virginia, and I attend Thomas Jefferson High School for Science and Technology.
I'm Dennis Gench. I attend Lexington High School in Lexington, Massachusetts.
Hello, I'm Daniel. I attend Limbrook High School in San Jose, California.
My name's Ethan Surya, and I go to California High School in San Ramon.
To start off, we'll be looking at Ascaris. It is a type of parasitic roundworm that infects both humans and pigs. The species that infects humans is called Ascaris lumbricoides, and it lives in the small intestine. There's also Ascaris sum, which mainly infects pigs. Doctors usually diagnose Ascaris by identifying its eggs in stool samples, as we can see in the micrographs presented in Figure 2. It also spreads through what is known as the oral fecal life cycle. This image shows the oral fecal life cycle of Ascaris. It starts when an infected person passes eggs in their feces. These eggs can get into soil, and then they can also be found on surfaces in public places in poor sanitation areas. If someone eats unwashed vegetables grown in that soil, they might swallow the eggs without knowing. Inside the body, the eggs hatch in the small intestine, and the larvae will travel through the bloodstream to the lungs. After a few days, they are coughed up, swallowed again, and returned to the intestines. There they will grow into adult worms that lay eggs, and a single female can produce up to 2,000 eggs in a single day. Those eggs leave the body in feces, and the cycle continues.
Specifically, ascariasis is the infection that's caused by Ascaris. It's one of the most common worm infections in the world, and especially in places with poor sanitation. Ascariasis affects about 10% of the global population, and its symptoms include abdominal pain and malnutrition and stunted growth in children. It's especially widespread in tropical and subtropical regions, and in fact, Ascaris eggs are often present on everyday objects like door handles. Children are especially more vulnerable to the harmful effects of the infection, and these effects can have long-term consequences on health and education. One of the biggest issues in treating Ascaris worldwide is drug resistance, because mostly current treatments aren't specific. So mass drug administration normally uses broad drugs like albendazole and mebendazole, which are used for many parasites, not just Ascaris. These drugs target specific proteins, but resistance to them is growing. There's also a growing resistance in animals with general drugs like moxidectin. The problem is most drugs don't target Ascaris directly, so the parasite is adapting and relying on general treatments is letting Ascaris and others evolve, making future infections harder to treat. Growing drug resistance to ascariasis treatments leads to prolonged infections in the host, worsening health issues, increased risk of co-infections, and placing a greater burden on the body's immune system.
So, why is Ascaris special? Well, as Ethan said just now, Ascaris is a very widespread problem. It affects 10% of the world. But the reason why Ascaris specifically is so special is that humanity has studied Ascaris for a really long time. It's actually a model organism for molecular biology. Look at this figure. It's from the 1888 year. This is over 100 years ago. Some of the earliest pictures of chromosomes that we have are from Ascaris. In addition, female worms lay a ton of eggs, as Teju has alluded to earlier, and the extracts from the eggs contain everything necessary to perform both cis and trans splicing. Now you might be wondering, what are those? Well,
Yep, and cis and trans splicing. In cis splicing, which is when normal humans and higher eukaryotes do, is when you have an exon to exon junction with an intron in between. Usually, for example, in exon 2 and exon 3, you can have an intron between, and this intron is cut out with a spliceosome. And then the exonic junctions are come together and the lariat is formed as a byproduct. However, in transplicing, which is what lower eukaryotes and like the Ascaris parasite does, is that you have an sLRNA binding to the first 5' acceptor of exon 1, and the byproduct of this is the Y branch. And if we can target trans-splicing without inhibiting cis-splicing, we can effectively inhibit the effects of the Ascaris parasite. And then using these exon to exon junctions, uh, we created these and inserted them into a plasmid with our hypothesis reminder, which we need to quantify trans splicing and mRNA processes.
Alright, so our hypothesis is that we can inhibit trans splicing without affecting cis splicing. As Denise just explained, by inhibiting trans splicing specifically, we will have found a way to specifically kill the parasite without harming people. Thus, our aim is to develop an RT-qPCR based screening assay in order to quantify trans versus cis mRNA processing. After all, these two processes are quite different, so we want to see what the baseline for each process would be.
And then this is a quick schematic to show where our actual research went through. So now we're going to start with our circular plasmid, in which we can see on the next slide um, where we inserted these exonic exonic gene sequences that we created into this plasmid with this AMP resistance that now Teju will talk on.
Next slide.
Yeah, next slide.
Now that our plasmid has the ampicillin-resistant gene, we introduced it to E. coli using bacterial transformation. We used the heat shock method to help the plasmid enter the cells. Then we grew them on plates with ampicillin. We did this to make sure only the bacteria with the plasmid would survive. So after growing it overnight, we picked one of the colonies and grew it in a liquid culture to get more plasmid for the next steps. After we grew the colonies, we picked a few and did our MIDI prep step. The purpose of a MIDI prep is to extract the plasmid DNA. We used buffers to break open the cells, then we passed the solution with our plasmid through a column that was positively charged. The negatively charged plasmid would then bind to the positively charged column, while proteins and other cell debris will wash away. In our final step, we used another buffer to release the plasmid, giving us clean DNA for our next step.
So our DNA plasmid is shaped like a closed loop, which is shown in the slide. Essentially, we need to cut that circular DNA to make it linear. This process is called linearization. Specifically, we use the enzyme EcoR1, and it cuts DNA in a specific sequence. Once the sequence is found, EcoR1 will cleave it at that spot to open up, so the circular shape is linearized. If the plasmid is not linearized, then the polymerase will continue to go around in a circle instead of stopping during transcription. And we confirm that the linearization was successful through running a gel electrophoresis. So after linearization, we can now move on to in vitro transcription. So in vitro transcription is essentially a lab protocol that is used to synthesize RNA from a DNA template. Next slide, please. And this process typically involves a DNA template that contains a promoter sequence, RNA polymerase, which is an enzyme that builds the RNA, and NTPs, which is a type of nucleotide molecule that builds the RNA also, and buffer. Essentially, the polymerase binds to the promoter, unwinds the DNA double helix, then moves along the DNA and adds complementary RNA molecules as it goes. And you can kind of see this on the slide.
All right, now moving into the denatured polyacrylamide gel and capillary electrophoresis step. You can see on the page gel on the left, we have a B cis and 3' acceptor. Now we need to separate the sizes of these acceptors and not to confirm if we actually have the right acceptors and the samples. So we have two buffers, the light blue and the dark blue buffers, which we already know the sizes of. And we already know that they're smaller than the samples of the 3' and the B cis. Thus, they should run faster on the gel and be beneath the samples which they were. So that confirmed our results. And then, our goal was to purify and check the RNA quality, which this step accomplished. As you can see, we confirmed that the RNA samples were indeed in the right size and they were pure after we extracted them. On the next slide, we can see a reminder that our next step is going to be the in vitro splicing assay. Now, in the in vitro splicing assay, we combined extracted pre-mRNA substrates with cell-free extracts derived from the nematode embryos. These extracts contained the necessary splicing machinery, which included all the proteins necessary. We incubated the mixture under specific conditions to allow splicing to occur. This is a critical step because in the in vitro splicing assay, allows the direct observation of the splicing process under controlled conditions in this glass environment, and it provides the essential evidence that the splicing machinery in the extract is functional and capable of catalyzing the cis or trans splicing reactions.
Alright, as a reminder, we seek to develop an RT-qPCR based screening assay in order to quantify the different splicing processes. However, you might be wondering, well, how exactly would you do that? Well, in order to do RT-qPCR, you first need to be able to make primers for trans and cis splicing. You'll see here that we have two diagrams showing the different splicing products that we expect from the splicing. You'll notice here that there are these short little comb-like sequences at the tops and bottoms of these products. These are our primers, and they're necessary in order to initiate transcription for qPCR. You'll notice that the primers go over the gap where the two different pieces of the splicing product connect. This is because in order to ensure that we are only amplifying the product and not just the individual components, we needed to specifically recognize this cross-junctional area and bind to it only. Now, with this, we will be able to engage in our experimental design. So you'll see this table to the left here as three different scenarios. One is positive positive, which means that our test has both RNA and the extract. The second scenario has no RNA, which means that nothing should happen in theory because there's no RNA to amplify. And in our third scenario, there is no extract from the eggs. As previously mentioned, the extract is needed for the splicing in order to occur. So if the first scenario succeeds and we successfully manage to amplify, but both of the other scenarios fail, then we know that we have hit our target. To the right, you can see the two graphs to demonstrate our results. The cis splicing worked perfectly. Only in the scenario where both RNA and extract were present did we have significant amplification. In all the other cases, there was nothing. However, in the trans splicing graph, you can see here that the other two scenarios where there was no RNA or extract amplified significantly, which shows that our process for trans splicing needs to be adjusted in the future so that we can better get a better result. You also notice in the corner there's these two bars labeled NTC. These are our non-target controls. They have neither RNA nor extract in them. And so they're just there to make sure that nothing's going wrong. So here is our qPCR. So you might notice at the bottom of this graph, there's like a key to demonstrate what each line means. You'll notice that the third color is labeled SARS, and you might wonder what that is. Well, this is actually what's known as an endogenous control. It's a known quantity of SARS RNA that we put into our sample to use as a reference and compare with the other amplification that we see. You'll notice also that there's a horizontal line at the top there. This is our threshold. It allows us to figure out exactly how quickly we have amplified our samples. This is important to know if our samples are actually amplifying correctly, first of all, and also to compare how quick uh, cis splicing is compared to trans splicing. because the faster it reaches that threshold, the more splicing is occurring.
The ability to quantify both cis and trans splicing concurrently will allow us to develop inhibitors of trans splicing. And this kind of research really helps us explore new ways to target parasites by focusing on how they process RNA, which could lead to better treatments in the future. Transplicing is used by a number of nematodes, trematodes, and trypanosomes, including the causative agents of sicknesses like African sleeping sickness. It matters not just for human infections, but also for animals, especially in farming, where parasites like Ascaris sum can create serious economic issues. Since other nematodes infect pets and livestock, what we learn here could be useful for tackling a wide range of parasitic diseases, both in medicine and veterinary care. Thank you so much for listening to our presentation. Here are some acknowledgements.
Yeah, thank you.
Thank you.
Thank you.